The beta cells secrete insulin, and we know that if they do not secrete insulin properly, or if there are not enough beta cells, then we get diabetes. Glucagon-like peptide 1 is being used as an anti-diabetic drug. It is used for the treatment of type 2 diabetes patients, and I will tell you how it works, and I will show you some beautiful pictures. In the normal body, the beta cells are in a ready mode. That means they are not in the off mode, so to say, and when they are in ready mode, they can be triggered easily by glucagon-like peptide 1, for example, and the other hormones that are secreted from the intestine. This is a simple experiment where we are measuring concentration of free calcium in the cytoplasm of a normal beta cell. We depolarize the beta cells artificially by giving potassium chloride, and the ATP sensitive potassium channels of this beta cell is paralyzed by diazoxide. The channel is opened by diazoxide. So as you can see, when we depolarize the beta cell in this particular experiment artificially for the sake of experiment, there is an increase of calcium, and the calcium stays stable after some time. But if we give some cyclic AMP to this system, then you will see soon that there are dramatic changes in the calcium signaling. The reason we are talking about cyclic AMP is that glucagon-like peptide 1 and related hormones that are secreted from the intestine, they are called incretin hormones, and they mostly work through the cyclic AMP system. They increase cyclic AMP in the beta cells. Is in the next slide you can see there is dramatic change in the changes in the calcium concentration as soon as some cyclic AMP is given to the system. In this experiment we did not give glucagon-like peptide 1, we gave phoscolin, which is a substance that increases calcium, increases cyclic AMP in the cytoplasm. Calcium goes up, in fact it goes very high only for a short period of time, and then it goes down, and then it goes up again and goes down and goes up again and goes down, and it can continue for hours. This type of calcium increase is due to calcium coming from the intracellular stores, and we can show in this particular experiment that this is really so. If you give thepsigargin, which is a substance that empties the endoplasmic reticulum calcium stores, these calcium spikes are gone. So the calcium-induced calcium release looks like this. They are very high increase of calcium, and they occur periodically, almost at some regular intervals. This is a very important phenomenon in the beta cell signal transduction. This experiment which you are seeing here is done by a human beta cell, and here we have given glucagon-like peptide 1, and the protocol of this experiment was designed to show calcium-induced calcium release in the human beta cells. As you can see, there is a beautiful increase of calcium caused by calcium-induced calcium release. So in the beta cell, when the calcium concentration is low, then there is no insulin secretion, and if calcium concentration is high and keeps high, then there is potential problem. Either there is not much insulin secretion, or eventually the cell can die because of high calcium concentration. But on the other hand, if calcium goes up and down and up and down, then there is plenty insulin secretion. That is exactly what GLP-1 does to the beta cell. Thank you.